Hi everyone, my name is Laura, and today I'm sharing a study that identifies a novel gut metabolite hormone circuit involved in regulating dietary sugar intake. Here's the full citation for the paper. It was published in Nature Microbiology just this past January, so it's pretty new, cutting edge stuff, and I'm excited to share it with you. So the bottom line up front is that host microbiome interactions shape dietary sugar preference and intake via a novel gut metabolite hormone axis. The study primarily aimed to investigate how these interactions shape dietary sugar preference with the broader goal of identifying new strategies for managing sugar consumption and related metabolic disorders. And one of the key findings was that downregulated expression of free fatty acid receptor 4, or FR4, particularly in the gut, is associated with metabolic disease states. Traditionally, this receptor is known for its role in fatty acid metabolism, but the authors also identified its role in shaping gut microbiome composition and downstream production of metabolites. And they identified Bacteroides vulgaris and its metabolite pantothenate as key targets of FR4 expression that can directly regulate metabolic homeostasis. Overall, these findings are exciting because they reveal a novel regulatory pathway underlying sugar preference that could be targeted in future strategies for reducing sugar consumption and improving public health outcomes. This work is really interesting to me personally as I enjoy exploring the connections between different bodily systems and how those connections can be leveraged to improve our knowledge and overall health outcomes. So, as lifestyles continue to industrialize, we see more and more convenience foods popping up. And with that, we are consuming these foods and beverages at higher and higher rates. Here's just one statistic from a study that surveyed adults in 185 different countries to look at trends in sugar sweetened beverage consumption, or SSBs. And we've seen a pretty substantial increase in just sugar sweetened beverage consumption over a 28-year period. In alignment with these trends, we've seen skyrocketing rates of diabetes, both type 1 and type 2, as well as obesity. And I bring this up because obesity and diabetes are generally considered diseases of the modern age, and they are driven and exacerbated by many lifestyle factors. However, I don't want to spread the message that sugar and diet are the only factors at play. We know that things like genetics, physical activity levels, and socioeconomic status also play a major role. But sugar intake is a contributor, and it's worth exploring. Chronic diseases like heart disease, diabetes, and cancer are among the leading causes of death and disability worldwide, and high sugar intake has been considered an environmental risk factor for the development of these diseases. We know that consuming sugar-sweetened beverages and excess sugar in general contributes to chronic disease risk through a variety of mechanisms, such as weight gain, hyperinsulinemia, and potentially through dysregulation of the nervous system and our reward pathways. So we know that sugar preference drives excess sugar consumption. And in this case, sugar preference has a definition. It's a biological phenomenon in which animals favor sugar over artificial sweetness. By sugar, I'm referring to true sugars with calories, like glucose, fructose, and sucrose that we can metabolize for energy. Artificial sweeteners are low or zero calorie and aren't really used for energy. And so the goal of this study was to determine what drives this sugar preference. And you might be thinking intuitively that it's taste, right? Because sweet things taste good, and when we activate our sweet taste receptors, we trigger the release of dopamine, and that keeps us coming back for more. But that's actually not the full story. In fact, a growing body of evidence is showing that taste alone does not drive sugar intake. I've cited some work from Diego Bojorquez's lab at Duke University where they study this, and they and others have found that signaling within the gut plays a significant role, but it's still underexplored. And so that is what we're tackling today, this gut microbiological basis for sugar preference. So before we get into it, I wanted to briefly go over some key players that will be coming up a lot in our discussion today. I don't have time to define them all in detail, but please bookmark this slide to aid in your understanding. we have FR4 or free fatty acid receptor 4, GLP-1 or glucagon-like peptide 1, FGF21 or fibroblast growth factor 21, and pantothenate or vitamin B5. And to make this more accessible, I have included each of these icons on the slides where these molecules come up. So hopefully that aids in the digestibility of this work. So the experimental approach involved two model types. First, we'll focus on our human participants. This Mendelian randomization analysis had a nice sample size of almost 65,000 people, and this was pretty much a large-scale analysis of data obtained from genome-wide association studies. And the researchers were looking at the relationship between FR4 mutations and sugar consumption behavior and habits among populations. So this was more of a proof of concept exercise. Then we had 84 blood donors made up of type 2 diabetics and healthy controls and 60 fecal sample donors. And from those blood samples, the researchers quantified things like fasting blood glucose, GLP-1, FR4 mRNA levels, and some other important biomarkers. And using the fecal samples, they were able to analyze and compare the gut microbiome composition of these different subjects. So moving into the animal models, most of the data we'll be looking at today came from mouse experiments. And so these proof of concept diabetic mouse models, DM, DBDB, and NOD, were used for the preliminary experiments just to establish the relationship between FR4 expression levels and diabetic disease states. And this was a sample size of 6 per group. Then for the genetic knockout studies, there were quite a few different knockout mouse models, but we're going to focus on 3. So we have our systemic FR4 knockout mice, or FR4KO, our gut-specific FR4 knockout mice, or GKO, and our gut-specific overexpression mice, or GE. And the sample size ranged from 6 to 10 mice per group. One confound I wanted to point out is that all the mice used in this experiment were males. Honing in on this box here, these are the 3 main experiments that they did with the mice. So this first one is representing the dietary selection test, where they gave mice a choice between a high sugar diet and a high fat diet, and quantified the amount of food that was consumed to determine what the mice preferred more of. The second one here is a similar type of experiment. It's called a 2-bottle preference assay, where the mice have 1 water bottle, which just has plain water, so that's like your control or no treatment, and then a second bottle with water sweetened with a sugar or an artificial sweetener, so that would be like the treatment. And the researchers measured how much of each liquid was consumed to determine what the mice preferred more of. And then lastly, number 3 is representing the co-housing experiments. These were done to evaluate the effect of microbiome sharing on the experimental outcomes. And so the co-housing experiments had 8 mice per cage, so 4 knockout or overexpression mice, and then 4 wild-type controls for a total of 8. Each cage setup had 3 replicates, and co-housing was done for 8 weeks at a time. For the fecal microbiota transplant, or FMT experiments, these were done only in mice. And so the way it works is feces were collected from mice, homogenized, diluted in buffer, and then this supernatant was orally inoculated into the mice at a dosage of 100 microliters per day for 30 days. All microbiome analyses were done using 16S ribosomal RNA amplicon sequencing. The authors did not disclose the specific sequencing platform used, but it was provided by a third party, GeneWiz. Beta diversity analysis was conducted using UniFrac, principal component analysis, and principal coordinate analysis to compare the microbiome profiles between the different experimental groups. OTU clustering was done to group similar reads into representative clusters, and they used vSearch with a similarity threshold of 97%, which is pretty standard. And then finally, LEFC analysis was done to determine which microbial taxa were most significantly affected by FR4 expression. Finally, the metabolomic studies involved culturing the microbe of interest under anaerobic conditions. And then using this in vitro culture, the researchers performed untargeted metabolomic profiling and liquid chromatography mass spectrometry, or LCMS, to identify and quantify the bacterial metabolites present in this in vitro culture. And all statistical analyses done for the entire study used GraphPad Prism, and t-test, ANOVA, and Spearman correlations were used to assess significance. And in this case, a p-value of less than 0.05 was considered statistically significant. So figure 1 shows the results of those proof of concept experiments. We have here those 3 diabetic mouse models, and the researchers are looking at relative FR4 mRNA levels in the colon tissue and the leukocytes of these mice. And the mRNA levels correlate to gene expression. So the more mRNA, the more of these receptors are being expressed in the tissue. And we see across the board here that mRNA levels are significantly depleted in those diabetic mouse models. Next, we're looking at our human data. So again, this is showing the relative FR4 mRNA levels in our normal healthy controls versus type 2 diabetics. And you can see again that gene expression is significantly depleted in diabetes. Panel D is showing the relationship between FR4 expression and fasting blood glucose, and we see that as FR4 mRNA decreases, fasting blood glucose rises into that diabetic range. I've included this line here to indicate the clinical diagnostic threshold for diabetes. Next, these are the results of our first sucrose preference assay. So again, the mice had the choice between the plain water and the sucrose sweetened water. Sucrose is table sugar, by the way. And so we have our 3 different diabetic mouse models here. And you see across the board that they have a higher sucrose preference ratio compared to their wild type counterparts. So this is our first piece of evidence establishing that relationship. We know that these mice have lower FR4 mRNA expression levels and higher sucrose preference. Figure 2 leads us into the genetic knockout experiments. So again, FR4 is highly expressed in many tissues of the body, but in this case, they knocked out the gene entirely. And so we see in panel G that the systemic FR4 knockout mice preferred a high sugar diet significantly compared to the wild type controls, but there was no effect on their preference for high fat diet. And so the key takeaway here is that FR4 knockout increases dietary sugar preference, but does not affect dietary fat preference. Here we're looking at another sucrose preference assay with the systemic FR4 knockout mice. So these top panels are showing the experiment with our dietary caloric sugars. So we have sucrose, fructose, glucose, and dextrin. And across the board, the knockout mice have higher sugar preference ratios. But then when it gets interesting is down here in these bottom panels, we have our artificial sweeteners, aspartame, saccharin, and quinine. And there was no effect on their preference for these. There's no increase. And so this is our first clue, going back to that initial point, that taste is not the only driver of sugar preference, right? Because if taste was the driver, we should see an increase. But something in the gut is allowing these mice to distinguish between the real and the artificial sugars. Figure 3 shows the results of the intestinal FR4 knockout mice experiment. So remember, GKO is gut-specific knockout. And again, these top 4 panels are showing sucrose, fructose, glucose, and dextrin. Across the board, preference ratio is elevated. But when we look at the artificial sweeteners, there's no change. The experimental mice look just like the control mice. Now we're looking at the intestinal FR4 overexpression mice. So our GE mice have this FR4 gene upregulated in the intestinal epithelial cells. And again, these top 4 panels show our dietary sugars, but the result is now flipped. So we see that overexpression of FR4 actually drops sugar preference across the board for all of these dietary sugars. And in each case, they're considered statistically significant. But again, when we look at our artificial sweeteners, we see no difference whatsoever. Figure 4 brings us into the co-housing experiments. So again, the mice were co-housed. We had the knockout mice and the overexpression mice co-housed with controls for 2 months. So panel A is showing a sucrose preference assay. Here on the left, this is the GKO mice before co-housing, so sucrose preference is as high as it can be. And then after co-housing with a normal healthy control, sucrose preference drops down. So this is our first clue that the microbiome is playing a role in shaping this sugar preference. Panel B shows the same experiment, but with the GE mice. So before co-housing, sucrose preference is lower, and then after co-housing with a normal control, sucrose preference actually rises. Now we're looking at the effect of antibiotic treatment on the sucrose preference ratios. And the reason they did this is because antibiotic treatment is standard before a FMT experiment. It pretty much clears out the microbiota before introducing that inoculum, right? And so all the mice receive this antibiotic cocktail, and the researchers actually did a sucrose preference assay after the antibiotic administration, and they found that these differences in sucrose preference disappeared, right? So it almost reversed the effects, which is more evidence that the microbiome is involved here. Now we're looking at the results of the FMT experiments. So here in panel E, we see here in this control section, this is before FMT. So the control mice have lower sucrose preference and the knockout mice have higher preference. Then in FMT, they switch. So the GKO mice received feces from the control mice, and the control mice received feces from the GKO mice. And so we see after FMT that their phenotypes switch, and now the controls look like the GKO mice, and the GKO mice look more like the controls. Panel F is showing the same thing, but with the GE mice. So here is before FMT, and then here is after FMT. So the key takeaway from this is that the gut microbiota is involved in FR4 mediated alterations in sugar preference. Here we're looking at the differences in relative abundance of gut bacteria at the genus level. And this was achieved through that 16S ribosomal RNA sequencing. And so we see here our control versus the GKO mice, and then control versus GE mice. And you can see by the colors that some genera are increased or decreased in the different groups. The thing that sticks out to me the most here is this dramatic increase in bacteroids in the GE, the gut overexpression mice. And this next slide zooms in on that. So the researchers identified a total of 6 different bacterial genera that were sensitive to FR4 expression, and we see those genera here. And lo and behold, Bacteroides is the most sensitive genus. And so here we're looking at the fold change in abundance of these bacterial genera. So a deviation to the left is showing a decrease, and to the right is showing an increase in abundance. And so we see in those GE mice, we have this dramatic increase in bacteroids, and then we have a pretty large decrease in the GKO mice, right? This is suggesting that FR4 expression is positively correlated with Bacteroides abundance. And one thing that was worth pointing out to me is this dramatic increase in Lachnoclostridium in the GKO mice. Abundance of this genus in the gut is associated with inflammation in cancers typically. So it's interesting that we see this pretty dramatic increase in abundance. And it gets me thinking about, because again, the GKO mice are our metabolic disease models, right? And so if we're seeing this increase in this, potentially harmful genus, Could it shed light on how metabolic disease could predispose us to more inflammation or other health issues down the line? Here in panel J, we're just showing the results of a Venn analysis that singles out Bacteroides as the most sensitive genus to FR4 expression. And from here, the researchers identified two main bacterial species belonging to the Bacteroides genus, Bacteroides vulgaris and Bacteroides dorii. And they did a sucrose preference assay and measured fasting blood glucose in response to supplementation with these species. So we see the GKO mice are serving as a control, and then the GKO that received B vulgaris are our experimental group, and you see this dramatic drop in sucrose preference. They also looked at the difference between fasting blood glucose in these groups. You see a modest decrease in fasting blood glucose, but nothing crazy. And they did the same thing with B. dorii, but I did not include the data because this bacterium turned out to have no effect on sucrose preference or fasting blood glucose. So B. vulgaris is really the key microbe here. Now moving into the metabolomic profiling studies. So once the researchers identified B. vulgaris as this key microbe, they isolated it, grew it in culture, and then did metabolomic studies to identify which metabolites might be important in this pathway, right? Because one of the ways that gut bacteria exert their effects on our physiology is through their secreted metabolites. And so here we're looking at a heat map of the relative abundances of the 11 key metabolites that they identified in their analysis, and we see pantothenate sticks out like a sore thumb. It is very abundant at 12 and 24 hours, and then panel F is just expressing this in a different way. We see the relative amounts of pantothenate increasing over time. So zooming in on pantothenate, we're looking at its relationship with disease states. So here we're looking at serum pantothenate concentrations in the mice, and the GE mice have the highest levels, the GKO mice have the lowest levels, and the controls are about the same. Then we see serum pantothenate in our human subjects, and the normal healthy controls have about 150 picograms per mil, which matches what we see in the GE mice. But then when we get into early stage diabetics, we see those levels drop, and then in diabetes, they drop even more. So this is showing that negative association with pantothenate and metabolic health. So this is probably my favorite slide. Here in panel K, we're looking at another sucrose preference assay. And so the GKO mice are the controls, and then they had a group that they supplemented with pantothenate, and you see that their sucrose preference lowers quite a bit. Then here in panel E, they did the same thing, GKO as the control, and then they gave the experimental group liraglutide. Now, liraglutide is a prescription drug used for weight management, and it belongs to the GLP-1 receptor agonist drug class, same drug class as the very popular Ozempic. And these drugs are known to work because they reduce sugar cravings and appetite in general, and control blood glucose. And so that's what we see here, right? Liraglutide reduced the sugar preference. But these results are very similar. And so it's exciting because it starts to bring us into these broader applications of the work that gut-derived pantothenate can reduce sugar cravings almost as well, if not as well, as liraglutide. Here we're looking at the relationship between pantothenate and GLP-1. So we see this linear relationship. This is in the mice. We see as pantothenate increases, so does GLP-1. And we see a similar trend in our human subjects, although we had a larger sample size here, and most people tend to fall right here in this average area. But I'm curious about these outliers, right? Maybe these are those super lean phenotype people. Who knows? And then here, panel C is showing the effect of B. vulgaris and pantothenate supplementation on GLP-1 levels, and you see a modest increase in both cases. I wanted to point out that the researchers also did this supplementation on healthy control mice, and they didn't see the same effect So it suggests that therapies involving either B. vulgaris or pantothenate might be more suitable for people with FR4 mutations or deletions rather than the average person. So now we're zooming in on the role of FGF21 in this whole pathway. And prior studies have shown that GLP-1 receptor agonists like liraglutide promote FGF21 secretion by the liver. And it's also been previously demonstrated that FGF21 is a key regulator of FR4 expression. And so the authors wanted to further explore the role of this hormone in this whole pathway. So panel F is comparing serum FGF21 levels in our mice among control mice, the GKO mice, and then GKO mice treated with liraglutide. And we see that liraglutide treatment significantly increases FGF21 levels, even compared to the healthy control. And one possible explanation for this is that higher GLP-1 level, right? Because liraglutide is increasing GLP-1. We saw that in the previous slide, and we know that GLP-1 is a positive regulator of hepatic FGF21 secretion. So that's one possible explanation for this effect. And then in panel I, we're looking at the results of a sucrose preference assay done comparing the effects of FGF21 on sucrose preference. And for this, the authors actually constructed a recombinant protein containing FGF21 that they stereotaxically injected into the mice. So stereotaxic injection delivers a compound of interest to a specific brain area. And we know that FGF21 crosses the blood-brain barrier and communicates with the ventromedial hypothalamus, which is involved in appetite regulation. So they specifically injected it to that brain area for this particular experiment. And we see that FGF21 reverses the effect of that FR4 knockout in the GKO mice. So this is a nice summary slide just to tie it all together. Really, the key takeaway of this whole study is that gut metabolite hormone interactions shape sugar preference. And by gut metabolite hormone, I'm referring to, with this gut piece, it's really the intestinal FR4 expression as well as the gut microbiota. This metabolite piece is referring to the gut microbiota's metabolites like pantothenate, and this hormone piece is referring to things like GLP-1 and FGF21. And so all of these components are interacting together in this novel pathway to shape our sugar preference and intake. So this block is reiterating that FR4 is a negative regulator of sugar preference. And so in humans, we see FR4 mutations or inactivations will lead to increased sugar consumption habits. And then we see, through our mouse knockout experiments that deleting intestinal FR4 specifically leads to increased sugar preference. And the key takeaway of this block is that Bacteroides vulgaris and pantothenate or vitamin B5 are the key targets of this FR4 mediated sugar preference pathway, right? We know now that decreased FR4 or FR4 deficiency leads to lower levels of B. vulgaris in the gut and thus lower levels of pantothenate produced. This final block here, the key takeaway is that pantothenate can actually boost GLP-1 secretion, and that itself is inhibitory against sugar preference because it stimulates hepatic FGF21 production. And again, just reiterating, FGF21 crosses the blood-brain barrier and it acts in the ventromedial hypothalamus. And so this is kind of bringing this whole mechanism full circle from the gut all the way to the brain. And so I imagine that future research by this group may pick up here and explore this piece a little further. So again, the key conclusion from this study is that downregulated FR4 alters the gut microbiota, particularly the abundance of Bacteroides vulgaris and pantothenate, which modulate sugar preference via the GLP-1 FGF21 axis. And the key insight that we've gathered from this work is we now know that FR4 has more functions than just fatty acid metabolism, right? We know that it can actually shape gut microbiome composition and affect sugar preference and behaviors downstream, and as well as metabolite availability. And so altogether, this work is establishing a novel pathway that we didn't know existed before. And the broader implications of this work are that we now have a roadmap for future microbiome-based strategies for combating diseases like diabetes and obesity. And understanding these connections might also have implications in neurological and behavioral disorders. I've heard some things about sugar intake being a risk factor for neurodevelopmental disorders in children. So, I'm curious to hear what you all know or think about that, because I'm not very well versed in that area, but, this is a potential application as well. And of course, some of the limitations of this study I mentioned before. Most of the data was obtained from male mouse models. In fact, all the mouse models were male. And in addition to that, mouse models aren't the best models for microbiome studies, although they are used pretty commonly. So, we are lacking human data. I mean, these results are really impressive in the mice, but we need to see human results, right? And we also don't know the long-term effects of any of this because this was a short-term study. We would have to see, how do humans tolerate B. vulgaris supplementation? I mean, I think pantothenate is a little less risky because people take B vitamins already, but this needs to be explored. And then lastly, other factors that influence sugar consumption, like overall diet composition, physical activity levels, differences in gut physiology and digestion, sleep and stress. None of these things were explored, which is understandable because this was already a dense study, but these factors definitely need to be considered in future research. And this work leaves a lot of gaps in knowledge and questions open for discussion and further study. So these are just some for you to consider. Beyond obesity and diabetes, what other conditions might be exacerbated by excess sugar intake? And what are some potential explanations for that observed increase in lactoclostridium abundance in the GKO mice? How could the findings of this study be leveraged to develop microbiome-targeted interventions for metabolic and related disorders? And what are some potential limitations of translating these findings from mouse models to humans? Again, we discussed this a bit, but maybe you have some ideas on how future research could address these limitations. And given that pantothenate influences sugar preference, is it possible that other microbial metabolites play a similar role in sugar preference? My guess is yes, and that we just haven't discovered them yet. Curious to hear what you think, or maybe you've heard of something else that has a similar role. And lastly, how does FGF21 regulate sugar preference at the neurological level? And what experimental approaches could future studies use to further investigate this mechanism? So, circling back to where this paper left off, I would like to see, a follow-up study kind of bringing this pathway together because it's so new. I'm sure that more pieces of the puzzle will be uncovered as we move forward. And finally, here's my references slide. All the information I mentioned can be traced back to here. I want to thank you for your time and attention today. I hope you enjoyed this presentation. I hope you learned something new, gained a new perspective, and I look forward to hearing your thoughts and feedback in our discussion. Thank you.